Hi, my name is Piper Tonne, and I am the leader of the Gut Microbiome team at Berkeley Pharmatech. This presentation will go over what I have learned thus far, highlighting my group project along with some key takeaways. I chose this group because I'm super passionate about neuroscience, and through past internship experiences, I have developed an interest in neurodegeneration. I have also always been super into nutrition and healthy lifestyle habits, and I believe they have a strong impact on your overall well-being. When I saw that the gut microbiome was an option for the research topic, I thought it would be interesting to merge these two interests. So what is our literature review about? The title for our paper is The Role of the Gut Microbiome in Neuromodulation Therapies as a Potential Treatment Adjunct for Multiple Sclerosis. When initially brainstorming topics for the paper, we noticed that many of us were super interested in neurodegeneration and wanted to know how gut health can impact the function of the brain. We also know that the gut-brain axis is a very novel topic right now in the medical field. Regarding multiple sclerosis, right now many of the current therapies aim to slow the disease progression, while the gut microbiome could provide a way to reverse the demyelination process. We began our research process by diving into the interworkings of the human microbiome. As you may know, almost 70% of the entire immune system resides in the gut. The health of the bacteria is highly dependent on many external factors, such as illness, antibiotics, diet, fitness, etc. The composition of the gut microbiome is ever-changing and highly sensitive to chemical, nutritional, and immunological pathways. In our paper, we looked into probiotic species and how modulating their presence in the gut may play a role in cognitive function. Our other area of research focused on multiple sclerosis. MS is a demyelinating autoimmune disorder that disrupts the communication between the brain and the rest of the body via cell-mediated or type 4 pro-inflammatory effects. The reason we are interested in the connection between the gut microbiome and the immune system is because MS is a disease that progresses due to this autoimmune inflammation, which is primarily triggered by T cells and potentially B cells in the central nervous system. Therefore, regulating the components of the gut microbiome that play a role in T cell activation could prove to be useful in treating MS patients. While we were able to find a lot of information about the topic, the interaction between the immune system and the gut microbiome must be explored further to fully grasp the intricacies between the connections and pathways associated between the two. We decided to take a look into novel therapies that could provide another avenue of treatment, such as neurostimulation, more specifically vagus nerve stimulation. This is a very new area of study, and the studies are limited, but we found research supporting both invasive and non-invasive vagus nerve stimulation that has helped those who suffer from similar neurological deficits to MS. So moving on to my experience with this internship, I think that it has made a strong lasting impression on where my interests lie within medical research. It has allowed me to do something not many freshmen in college have done, which is research on a topic that I'm very interested about. I have noticed how passionate I am about neurodegeneration and finding new ways to approach treatment that doesn't involve invasive drugs. This experience has prompted me to want to learn more about biotechnology and the increasing role it has in medicine. Another highlight from my experience so far as an intern at Berkeley Pharmatech has been the connections that I have made from people all over the US who share similar interests as me. With school being online, it has been hard to make those lasting connections, but through our weekly meetings, I feel I've been able to make some special friendships. One thing that has worked very well for our team was how we divided up the work in the outline of the paper. Since there are 7 members in my team and 7 areas of interest that we wanted to research, we naturally all were drawn to one topic. When it came time to writing and editing our paper, we all wrote one section in the outline and then formed an editing rotation so we were able to review all sections. This editing rotation proved very useful. One challenge that our team confronted was how extensive to make certain parts of our paper. Since our overarching topic was the gut microbiome, we didn't want to steer too far away from this in researching MS. At first, it seemed like there wasn't enough evidence to support a connection between the gut microbiome and MS, but the more we looked into it, the more we found. We noticed that that the two topics can be connected through research regarding the immune system, as the gut microbiome is home to the majority of the immune system, which also plays a large role in the nervous system. Through looking at our paper from this perspective, it provided us with more clarity and guidance in research. One thing that I wish I would have known at the beginning of this internship is that to be a leader, it takes practice, and that there's definitely a learning curve. Throughout this internship, I became more confident in my abilities in leading my team and was able to be more comfortable. I learned that it is okay to ask for help with certain tasks and to use my resources for assistance. I also became much more efficient at time management and creating a strong work ethic throughout this internship. In making this knowledge transfer, I decided to get my team involved, as I have made some amazing connections with the members. Here are some key takeaways from the gut microbiome team.
Hello, my name is Deondre, and I'm a recent graduate from UCLA. I'm thankful to Berkeley Pharmatech for giving me the opportunity to build my confidence in academic knowledge as I pursue a career as a physician scientist. I'm grateful to my team and the individuals I've worked with, especially our director, Vandawan, for giving us the freedom to explore and build ourselves as individuals and leaders of the next generation. I will never forget my time here. Thank you, everyone.
My name is Nabiha Hassan. I am the innovation leader of the Gut-Brain Axis Group. My biggest takeaways from this internship were improving my time management and public speaking skills and learning the procedures of writing a scientific paper.
Hi everyone, my name is Sravya, and I think my biggest takeaway from this internship, aside from being able to work with such incredible and intelligent people, is that knowledge truly is power. Research and technology have the ability to save countless lives, and I'm grateful to have been part of a small step in the right direction.
Hi everyone, my name is Arya Reddy. I'm a biomed research intern at Berkeley Pharmatech, and my biggest takeaway during this internship was the importance of collaboration and feedback.
Thank you.